The Plenary Scientific Session is a highlight of the annual meeting where researchers present the highest caliber scientific abstracts.
The investigators showed is that with the post-transplant cyclophosphamide, there was much less graft versus host disease, but it didn't impact on relapse rate. So it was a safer transplant with excellent results, and it's probably a new standard of care for bone marrow transplantation. What these investigators did is they gene edited the cells to try and understand the basic biology. So the interesting thing about severe congenital neutropenia is the gene, when it's mutated, it makes a protein product, but the protein product, it's there in normal numbers, it just doesn't fold correctly. And what they showed is when they knocked out the gene, they could get around the defect. And this opens up a great potential for genome editing. The fascinating part of this study is that the CAPS patients, the catastrophic antiphospholipid antibody syndrome, the patients who have the most severe phenotype, they also had mutations in germline mutations in genes that regulate complement. So these patients, in essence, have a double hit: the antibody activating complement and then a gene underneath that makes them more susceptible. And this explains why this is a more severe phenotype. This is off the shelf, and it appears to be quite safe, and it will likely be much less expensive. So a lot of the problems with CAR T cells are overcome potentially by this bispecific antibody. It's a large study. There's 218 patients. It had a very high response rate. These are very refractory patients, and some of the patients had even failed CAR T cell therapy, and they were able to salvage them. The response rate was over 60%. Many of them had complete response rates, so it'll be very exciting to see what comes of this in the future.